All right, so we're gonna go ahead and get started. So now that we've got our timekeeper up here, so Krista Hopkins. So for the speakers, Krista is the one who's gonna show you how much time you have left, and 10 minutes max, so you are done at 10 minutes. We're gonna stay on schedule. So you get like a little card at 5 minutes, and then a card that says you have 3 minutes left, and that's about the time to kind of wrap up your presentation, leave some time for some questions. Then you'll get another 1 minute warning that's like wrap up the answer to the question, and then you'll be done at that point. So we'll work to stay on schedule. For the Q&A, if you have a question, just come up to the microphones. Feel free to line up if you want, so we can get through those pretty quickly. So welcome to Energy Week. I'm Brian Korgel. I'm director of the Energy Institute, professor in chemical engineering. I'm really excited about Energy Week. I think it kind of highlights and illustrates all of the things going on at UT in terms of energy. So we educate students. Students are our priority. That's what we do. We've got our classes, our research groups and all of that. But UT is also a regional convener for expertise, so industry partners, philanthropic organizations. We've got an amazing network of alumni who are leaders in the energy industry, and I think you'll see that in the program this week. UT has over 450 faculty and staff who do research in energy in our 19 schools and colleges all across campus. So it's an amazing group of people doing really interesting work. So I'm excited about this morning's session. It's all focused on students and student research, 10-minute presentations. We'll hear progress on the Strategic Seed Research Program that's going on, so 12 different projects. And I said 10 minutes each. Really looking forward to it. So the first talk is going to be on distributed and electrified green ammonia production using plasma catalysis, presented by Vivian Lee and Varanasi Sai Subankar. I assume you all are here. Yeah, great. And is it two of you presenting or just? Yeah, okay, come on up. Do they control this up here? Somebody? Okay, there you go.
Thank you. Okay. Good morning, everyone. Today I'll present our research on green plasma productions using plasma catalysis. It's a collaboration effort between 3 faculty members and 3 students here in UT Cochrane School of Engineering. Before I get into the science, I want to acknowledge the support of Energy Institute as well as our industrial sponsor who make it possible for us to make the research, pursue the research over the past year. So this project was motivated by reimagining how we can make and utilize the plasma. Traditionally, Haber-Bosch is the main process to produce ammonia in the industry scale. As we can see in the plot here, ammonia is flavored at the low gas temperatures. However, in the industry scale, because we want to make it quick and in a large amount, so we need to increase the temperatures. So therefore, the industry scale conditions will be extreme at nearly 800 Kelvin and 100 bar. So therefore, Haber-Bosch process yearly consume about 2% of global energy. So instead of that, our work is focused on explore how plasma energies, electrical energy, and catalyst material combine to synthesize ammonia at mild conditions. So why is it beneficial? So if we can activate the nitrogens with the electrical energy, we can synthesize ammonia at room temperatures and atmosphere pressures. So what that mean? We can synthesize ammonia on site, which can reduce the transportations distribution network. And if we use the electrical energies with sustainable sources, we can lower the carbon footprint emissions from ammonia productions. So over the past year, we have designed the reactor we have designed and optimized the reactor to investigate the synergy between plasma excitation and catalytic material. So here, so this is the lab scale reactor that we develop here at UT. So this which can operate at ambient conditions. So it's figure up to 100 SCCM flow rate and 10 watt for input power. So within the reactor, the mixture gas of nitrogens and hydrogens will flow at 1 atmosphere, and where the filament spark discharge will apply to activate the nitrogens before it interact with the electrode catalyst to synthesize ammonia. For plasma system, usually the electrodes used at electron emitter, but our focusing is we think maybe we can use if we can use the electrodes at the catalytic material to try chemical reactions and have the extra values products. So the plot right here is the density functional theory, which simulates how ammonia productions with different catalysts in plasma. And as we can see here with the red and purple curve, if we don't change the temperatures gas much, but increasing the excitational vibration excitations of nitrogen molecule, we can definitely enhance the production rate for ammonia. So our research right now initially start with copper, which is common electrode, but moving forward, we will investigate the nickel, which is showing the significant ammonia production here. And this bottom plot here is our experimental data for tungstens and copper electrode. As we increasing the input energy, the ammonia yield is also increasing. And at 16 EV per molecule for copper electrodes, we yield about 5% of ammonia, which is what I want to highlight here is we can make it at near room temperatures, 1 atmosphere, and temperature variational is 3000 Kelvin. And where compared with Haberpos, which can yield up to 20% in volumes, but at much more higher conditions and extreme energy costs. So the critical metric we use is energy yield, which is how much energy we need to produce 1 gram of ammonia. So what we're looking for is we need it's high percentage ammonia yield and high energy yield as well. So using the pool spark discharge combined with the copper electrodes, I think we are in the upper range of what we have compared with other plasma-based approaches. And we decided to test the nickel electrode, which was so offer the higher productions from these simulations. And so the key question is, what is the acceptable energy cost where we can synthesize ammonia on site?
Thanks very much, Tui. Hello, everyone. I'm Dr. Harry Canada. I'm not a student. I'm a lecturer here. It's good to see some students in the room. For the last slide, I'm just going to step us through some of the sort of techno-economic implications of what this research shows us. So the main thing that Tui sort of highlighted there, which is relevant to the viability of this process, is the difference between the energy required for the Haber-Bosch process and our process. So in the top left, you can see that we are roughly operating at about 80 gigajoules per ton of ammonia, which is approximately 10 times the energy cost of Haber-Bosch. So we're not going to compete on that industrial scale process. We see there are really interesting opportunities in terms of making this process viable if we optimized across different aspects. So one aspect of that is distributed production. So we could be in a mountainous region, which is very hard to get ammonia to ordinarily. And that might be a really productive way of proceeding, where we're optimizing on the distributed nature as opposed to the overall energy efficiency. There are other aspects. Modularity is another idea. We could incorporate it with different inputs in terms of the hydrogen, where it comes from, electrolysis. And then scale is another interesting thing. We might be able to move towards pocket ammonia. I've invented that term, but you wouldn't want to do this in your pocket, but that could be an interesting direction. And then there's considerations of reversibility. So I will leave it there because we're very tight on time, and we'll move to Q&A. Thank you very much for listening to us.
If you have any questions, come on up and ask them. All right, I'm gonna come up with a question. As y'all are formulating I know it takes a little while in the morning. You're like, hey, yeah. All right, Michael, come on. Use the microphone though, yeah, because we have. So I don't know if we actually. Sorry, thanks.
This is cheating because I'm part of this project, but I have a question. Harry, for the slide, if you improve the output from 10% yield to 80% yield or something, would that just give you 1/8 and that gets you closer? Like, what are some of the pathways to either reduce the cost or improve the output that you can think of that make it more relevant?
In a sense, I think that's probably Tui's department in terms of catalysts and other aspects for improving that kind of thing. So maybe
can you improve the yield from 10% to something higher, I guess is our question for you, Tui.
I'm sorry?
Can you improve the yield from 10% output to like 80% or something else?
Yeah, if we...
Because that would fundamentally change Harry's analysis.
Yeah, because our project now is just like input small watt like energy, just like in order of 10 watt or lower. So if we input more like energy in, we can increase, hopefully we can increase the yield of ammonia. Yep.
All right. Okay, we'll move on to the next presentation. Thank you.
Okay. Yep. Thank you.
All right, so next we've got Akshat Singh, enhancing the durability of electric catalysts for the oxygen evolution reaction through strong metal support interactions.
Hi everyone, I'm Akshat from the Risasco and Milaran groups at UT, and I'll present my work on strong metal support interactions for electrocatalysis, more specifically for hydrogen fuel cells and electrolyzers. And so to start with, what do I mean by strong metal support interaction, or SMSI? It refers to the phenomena where a metal particle gets encapsulated by the support upon high temperature reduction, as shown in the schematic here. And while this has been well explored for its effects on reactivity and stability in thermochemical systems, it's not as well explored in electrochemical systems. And so this is what we wanted to explore. Using this model system of platinum supported on anatase TiO2, and then we reduced it at high temperature, 600 degrees Celsius, and saw encapsulation of the platinum nanoparticles with a very thin overlayer of TiO2. And so through my presentation, I'd like to answer 3 specific questions. Firstly, is this SMSI overlayer even stable in electrochemical conditions for it to be of any use? Secondly, how does it affect the stability of the supported platinum nanoparticles under applied voltage and pH? And thirdly, how does it affect reactivity? So in order to test the electrochemical stability of the SMSI overlayer, we performed 3 experiments. Firstly, we looked at carbon monoxide stripping voltammetry, which is a technique that is used to quantify the number of accessible platinum sites to an adsorbate, which is carbon monoxide in our case. And what we saw was for the encapsulated version, which is the red curve at the top, the CO stripping peak area reduced significantly compared to the other catalysts, suggesting that the overlayer is still stable under the applied potential and pH. Then we performed some accelerated stress tests, or basically we cycled the catalyst under in EChem conditions, and we looked at the catalyst post EChem using TEM and EEL spectroscopy, and we could still observe the overlayer post electrochemistry. And through EEL spectroscopy, we could look at the titanium edge as we went from the support to the interface to the platinum, suggesting that the overlayer is still intact even after long-term cycling tests. And so how does it change the stability and reactivity of the supported platinum nanoparticles towards fuel cell and electrolyzer reactions like oxygen reduction or hydrogen evolution oxidation? So firstly, talking about stability, we saw that we looked at the hydrogen under potential deposition area, which is this y-axis, as a function of the cycle number, and we saw that for the encapsulated catalyst, the reduction in the accessible surface area was lesser for the encapsulated catalyst compared to the non-encapsulated, suggesting that the overlayer is kind of fixing or immobilizing the platinum nanoparticles on the support, thereby improving stability, preventing dissolution or agglomeration under EChem conditions. And then we also saw that there was a reduction in the oxygen reduction activity for the encapsulated catalyst. However, the hydrogen evolution oxidation reactions activity stayed unchanged, suggesting that the overlayer can probably act as a size-selective membrane of sorts, where it's allowing the permeation of smaller molecules or atoms like proton. However, it's hindering the accessibility of larger molecules to the platinum that's under the overlayer. So, yeah, thank you. That's all.
All right, questions? Yeah, come on up.
So my question is, so is this unique to platinum and titanium oxide, or is this something also seen with other types of?
Yeah, that's a good question. So you can observe this phenomena with other metals and supports. One thing is that we want the support, which is TiO2 in our case, to be a reducible support. So it could be, let's say, niobium oxide, which is again reducible, but something like aluminum oxide cannot be used to observe this phenomena because it won't reduce even at high temperature. So that's one. And then you usually want the metal to be a high surface energy metal. So something like platinum, palladium, or even iridium can be used to see this phenomena. Cool. Thank you.
All right, great. Well, next talk is Mingfeng Xie, who's going to talk about innovating green hydrogen production, synthesizing high entropy alloys via bubble printing for enhanced electrocatalytic performance.
Okay, so hello everyone, this is Minfeng, and I'm supervised by Professor Yubin Zhang, and also this project is collaborated with Professor Simon Humphrey. So the title is Innovating Green Hydrogen Production and Synthesizing High Entropy Alloys for Enhanced Electrocatalytic Hydrogen Evolution. And just like title said, we are exploring the novel materials for hydrogen evolution reaction, because conventionally the best catalyst for this kind of reaction is palladium or platinum, but they are very expensive, so therefore the practicality is limited. So that's why we are looking into the high entropy alloys. And also, high entropy alloy has some good property because of its nature. It's usually composed of 5 or more elements inside its lattice, and it offers some properties like compositional flexibility and atomic configuration, and also structural stability due to its high entropy nature. So people usually use it in energy applications such as fuel cells, battery, and also electrocatalysis. And as for the synthesis parts, conventionally there are a lot of limitation. As you can see here, most of the synthesis require large amounts of solvents or energy, it causes a lot of waste. And also because they are usually powder form products, so people usually need additional steps for working electrofabrication. And we can address this limitation based on our synthesis. We call it bubble printing synthesis. It's like a laser direct writing method. So we can direct printing high entropy array on the substrate, which can be an electrode like FTO glass. And this synthesis is under ambient condition. And also because it's a printing process, it allows the design of the catalyst pattern. So in this slide, I want to exhibit the mechanism and also some demonstration. So for the substrate we use, we can use the glass. It can be FTO glass, like conductive glass. And we will put some gold nanoparticles on it. We usually call it gold nanoislands. And it's a plasmonic substrate. So when we irradiating the laser on this plasmonic substrate, it can generate photothermal effects to increase the temperature in the solution on this substrate. And because of that, we can induce a microbubble here. And because of the surface tension gradient and the temperature gradient induced by these photothermal effects, it can generate a Marangoni convection flow to guide the metal ion in the solution to the region below bubble. So it causes a metal ion accumulation effect. And also, because of the light irradiation, it can induce a localized reduction effect, like the hot electron from the plasmonic substrate will induce the reduction of metal ion here. So with these two effects, we can successfully synthesize high entropy arrays, and just like this video shows. Yeah, and here's the pattern we try to print. And on the right side, we have demonstrated characterizations for these septenary high entropy arrays. So in the first part, we do some SEM image and index mapping. And we can see that in this line structure, we can have like 7 elements inside. And also we do some transmission electron microscopy to see the crystalline information. So from STEM image, we can see that the structure is kind of composed of a lot of small polycrystal, and it shows the polycrystalline nature of my material. And also in TM, we can see most of parts are crystalline. Maybe a little part is amorphous, but most of them are crystalline. And from the fast Fourier transfer, we can see that the pattern looks like a fast center cubic. So we can kind of confirm that my material is like these 7 metals sit in the FCC lattice. Yeah. And the final part is the catalysis for hydrogen evolution reaction. So the material I use for this reaction is this, iron, rhodium, palladium, and platinum. And this is the pattern I use for this kind of reaction. Here is the system, graphite rod as a counter electrode, and also reversible hydrogen electrode as a reference electrode. And here's the photometry result. So I did 3 repetitions from 3 different batches of this material. And basically, we can see that from the average of these 3 trials, we can see that the onset potential is slightly positive, showing that because we are comparing with the reversible hydrogen electrode, which is using platinum as the catalyst. So with this result, I think I can say that the activity is close to platinum or even better than platinum. And also I'm offering the overpotential value has this certain specific current density, and we can see the value is pretty low. And I'm just comparing this value to other similar works like this are also focusing on the high entropy arrays in acidic hydrogen evolution reaction. And we can see that our overpotential value is the lowest. And also we also kind of lower the normal metal percentage in our work, which showing that our work is pretty promising. And for some future direction, we can provide some theoretical calculation from density function theory and maybe just calculate the absorption energy to proton, and we can further verify this result. Yeah, and that's all.
All right, questions? So as y'all are formulating a question, I have one. So in terms of scalability for your process, is there a way to, I don't know, scale up the method to make a larger amount of catalyst, for example, or have you thought about that, or in a 3D system?
For scaling up, I think it would be it will also become a time-consuming process to scale up. We can do larger area printing, but yeah, I think it will take more time to do it. So maybe this technique is more suitable for screening process to screen which composition has a good performance in this kind of reaction. Yeah.
So for the screening, have you thought about trying to combine like AI methods or anything in terms of for materials discovery, or is that something y'all are thinking about?
And that's a good point. I mean, I didn't think about that part because if for machine learning, maybe it would be hard because machine learning needs a lot of database to train the model. So yeah, I'll look into maybe AI has something can do here. Yeah, thank you.
All right. Well, if there are no other questions, thank you. All right. So we'll let you know all the talks are being recorded and will be posted on the YouTube channel and everything later. So you don't feel like you have to video the whole thing right now or anything like that. And also the speakers will be around. So the next talk is by Cameron Carter, De-risking carbon capture with amine solvents using high-resolution mass spectrometer methods. So as Cameron's coming up, there's still time for audience favorite voting on the posters next door. So the posters will be up today. We had a great poster competition yesterday. There's real money at stake for the students. So we want the best posters to be selected, and there's an audience award. So feel free at the breaks and lunch to go over and take a look at those and vote on your favorite. The students will appreciate it if you vote for them. So thanks.
Hello, everybody. My name is Cameron Carter, and I'm a staff chemist working in collaboration with Dr. Fred Klossman, Ian Riddington, and Pavel Mishtov in support of the 2024 Strategic Energy Seed Grant Program. Today we'll be discussing risk mitigation and amine solvent-based carbon capture using advanced mass spectrometry. So this is our absorber stripper model. If you look over here on the left, this is the absorber. So piperazine solvent is going to be flowing countercurrent to flue gas from either a coal or natural gas power plant. This flue gas contains CO2, oxygen, NO2, and some other gases. CO2 is going to react with piperazine. CO2, is this on? CO2 is going to react with piperazine to form piperazine carbamate and store CO2 aqueously. And then oxygen is also going to enter the system. This becomes an issue as oxygen is then going to oxidize piperazine into some form of degradation. Following the absorber process, we then move on to the stripper. So this is a temperature swing process in which this newly reacted piperazine and CO2 will then dissociate in the stripper, resulting in piperazine being recycled into the process and CO2 moving on to the next stage. You'll be accumulating some sort of degradation, both oxidatively in the absorber and thermally in the stripper. And so liquid and gas phase analysis is incredibly important for understanding what our degradation products are in those two phases. I think I'm having some technical issues. All right, so with the UTC grant, these are our three primary objectives that we worked on. So first, we worked on helix separation with Dr. Ian Riddington. This was done using a QTOF MS. Our first objective, we were able to improve our chromatographic separation in our liquid phase. This is dominated by about 30 weight % piperazine. And so that's a major portion of the solvent. So with this helix separation, we're able to improve our ionization efficiency, and we increased our detection capabilities with this. Furthermore, with Dr. Ian Riddington, we were able to observe iron chelation using QTOF-MS. So one of the degradation products we've begun to observe in our degraded piperazine solvent is amino acids. So it's thought that amino acids are able to solubilize iron, and this is going to result in enhanced amine oxidation and equipment corrosion. So using QTOF-MS, we're able to develop a method that was able to observe these amino acids chelated or complexed with dissolved iron, and we did this using EDTA and iron as a base model. Finally, with the help of Dr. Pavel Mishtov, we're able to use his Vocus PTR TOF to monitor emissions. So by doing this, we're able to test headspace gas of degraded piperazine solvent under controlled conditions, and this allowed us to monitor volatile degradation species. With this information from our both liquid and gas phase analyses, we're able to estimate Henry's constants, and these will be very useful in predicting CO2 capture system emissions. So back to helix separation. So in using QTOF-MS, it uses two portions of the instrument. This is going to be the LC, or liquid chromatography portion. This will be the MS, or mass spectrometry portion. In typical separation, polar species will elute very quickly, and non-polar species elute much later. So our solvent piperazine and many of its degradation products are highly polar. And so therefore, once you send the solution to the MS, you get a huge smattering of peaks, and you get very little separation, which makes ionization efficiency very low, and therefore makes detection very difficult. Another thing is that piperazine, being one of our most dominant species, is going to take up much of the competitive ionization efficiency from the MS, also reducing detection. So with Ian Riddington set up what's called a helix separation. This is going to be hydrophilic interaction chromatography. And so this basically works in reverse of traditional chromatography. So non-polar species will elute much earlier than polar species. So this makes ionization efficiency much better because we get separation of all of our degradation species, PZAA, HED, PZAA, PZ, as well as many others, without having piperazine be the most dominant species, absorbing a lot of that ionization from the instrument. This makes our detection much better, and then also allows us to see smaller species that usually wouldn't be able to see. So Ian Riddington, again, helped us with his QTOF-MS. This is EDTA and iron in our model system. So this was using helix separation, and we were able to separate EDTA from iron chelated EDTA. Typically, with most chromatographic methods, this would not be very easy. However, helix separation allowed us to separate these two peaks for identification. And so if I were to scroll over this peak, we'll see this. So this is going to be the MS portion. Most metals, when they undergo MS detection, will produce this particular fingerprint. Each metal has a different identifying fingerprint that's associated with it. So this is how we're able to confirm that EDTA is chelated with iron, and this isn't just some species that's somehow abducted, or we know for sure that this is going to be EDTA chelated with iron. So finally, with Dr. Pavel Mishtov, we were able to use his Vocus PTR TOF, and this instrument's able to continuously monitor volatile organic compounds down to about 1 part per trillion. This technology has the ability to detect thousands of compounds simultaneously, and this is all done in the gas phase. So using this instrument, we're then able to relate what we had already known, which is as time goes on in the process, piperazine begins to degrade heavier. And so we know in the liquid phase degradation products go up, and now we can confirm in the gas phase that our degradation products go up. So with the help of the Vocus PTR TOF, we're then able to quantify our gas phase emissions and relate those to our liquid phase emissions and develop what's called a Henry's constant. This allows us to measure volatility of our species. So here are top species that we had found, some of our degradation products, and here are their Henry's constants. As they go down, they become more volatile. So in the future, we would be looking at mitigation methods that would most suit mitigation of some of these species down towards the bottom. I'll go ahead and take any questions. Thank you all.
Hi, thanks for the presentation. My question is about the future direction you were just talking about. Have you considered ranging the amount of iron ions that are in the solution to see the effect of that concentration on the degradation rate, and then what you would do to mitigate that, like either add antioxidants or chelators to the actual carbon capture solution?
Well, yes, we've looked into kind of scaling how iron is going to
Maybe you use the microphone? Yeah.
We've looked into scaling how more iron has become more of an issue. In this particular research, we did not do that, but we do know that over time in the process, iron does increase, and this is likely from, again, corrosion of the hardware. So more research is needed to really nail down how this is going to affect emissions, but we do know that iron is increasing over time in the system. And then you had a second part, I thought.
Yeah, what you're going to do about it, basically. What
Mitigations-wise?
Yeah, mitigation.
We've thought of additions of some sort of amino acids or other chelating compounds. However, we do not know the effects that would have on solvent degradation. We'd likely expect that solvent degradation would increase if we were to add something like that. Thank you.
Any other questions? I can also come to you if this is too intimidating to come up here. Happy to do that. I have a question. So I don't know that much about these systems, but do you control the pH in your? I mean, if corrosion is an issue, is there a way to control pH, or are you stuck at like really basic pHs just by the nature of using the amines?
Right. So this amine is highly alkaline, or pretty alkaline. So it's also a massive portion of the solvent. So it's going to stay very alkaline regardless of what we really do to it. Mitigating corrosion, one of the issues is because we've got this absorber stripper system, the formation of these degradation products is what is causing or likely causing this corrosion. So modulating pH could be a way to work that out in the future. However, we do know by adding, if we were to, for instance, make the solution more basic, you'll then begin to dissociate piperazine from CO2 and reduce efficiency. Right now, we've got about a 98% carbon capture rate with the system as is.
Got it. Okay. Yeah, come on up.
Good morning. I was just wondering if through your carbon capture system, if you're able to set that up in just like cities and stuff like that to be able to absorb the CO2 to capture it and then turn it into the CO2 in more of a purity with this system.
I'm sorry, so captured in cities, like with like a power plant?
Yeah, just like in the air.
So in kind of like a direct air capture kind of system?
Right.
No, this is not scalable. Well, I don't know. But this system is primarily driven for high CO2 environments. For instance, like our flue gas station. However, I wouldn't see why not if we could set it up that way.
Yeah, I was just looking at it for maybe redesigning it for something else, but also whenever you're coming off that, what do you do with the CO2? Do you put it in a container or?
So sequestration. So following the association of piperazine, CO2 would then move on to the sequestration portion, which in some cases people are mineralizing, other people are using doing other things with CO2. But there is an end product, and that being highly pure, or not highly, but fairly pure CO2.
Yeah. So how can I get that?
You want it?
Yeah.
Well, then we can set something up.
No, I want it because I'm using SCO2 in a geothermal system, so I'd like to.
So then, for instance, it could probably be shipped off to you for your next stage. My field is not sequestration, so I won't field any questions that are related to what's going on that half of the system. However, we could probably set something up.
Yeah, I want to use it as a working fluid in SCO2. So that's why I was asking.
Okay.
So maybe I'll get with you and see if we can get some from you.
That'd be great.
Thanks.
Yeah, thanks. Thank you.
All right.
Thanks
Thank you
much.
all.
Yeah. So speaking of CO2, like we've got a great energy symposium today that's going to go deep into the weeds. Sanjay Bishnoi, who's actually proud alumnus of our chemical engineering department, got his PhD here and doing amazing. We've got a panel also on carbon capture and storage, so a lot of that. And we have another talk on that. So the next talk is Selfinaz Velioglu, increased carbon sequestration and storage by accumulation of plant root biomass and microbiomes.
Hello, I am Serpilnaz. I am a PhD student in plant biology at Dr. Chan's lab. Today I'm going to talk about our project, which is increased carbon sequestration and storage by accumulation of plant root biomass and microbiome. Our rationale is that atmospheric carbon dioxide is rising up about 2 parts per million every year. And in this figure, you can see that in 2020, it reached about 412 ppm. Plants help capturing these carbon dioxide emissions about 25% via above ground and root biomass. Also, agriculture soils may offset the 10% of global greenhouse gas emission via carbon farming. Our goal is to understand the role of the specific plants, the genotype, and microbiome interactions in the soil below ground carbon storage using the hybrid plants and their parents. Here we can see the simple diagram that atmospheric carbon dioxide is taken by plants through photosynthesis. It leads more root biomass and root exudates that feed soil microbiome. Together, root biomass and soil microbiomes can help store the carbon. So this brings us to our next point, that how hybrid plants can play a special role through the phenomena hybrid vigor. The hybrid vigor means that here here, with the distinct parents, Arabidopsis thaliana and Arabidopsis aracena, you can make a crosses and you will get the F1 hybrids that are bigger than their parents. Arabidopsis is small flowering plants that are used in biology studies because they are well studied and they have the genome assembly. Also, we can see the same pattern for the maize. Here again, you can see one of the parental lines, B73 and MO17. When we cross them, we can see that their hybrids are grow better than their parents. Also, we can see the production increase. Based on these two model system, we have 3 objectives in our project. The objective 1 is using the hybrid parents and using their root biomass to decide how much carbon dioxide is converted in Arabidopsis and hybrids maize. The second objective is that using this mechanism for increased growth of plant roots and microbiomes in Arabidopsis. Also, the third objective is that we can characterize the metabolites and small molecules for crop improvement. Today, I will be talking more about the first objective for the maize plants. Here is our experimental approach. So we did planting for maize plant of B73 and MO17 as well as their hybrids plants. We did the measurement for plant height, plant width, also the root biomass. I also collected the soil and harvested the roots. We also have the rhizosphere sampling. Those are the root attached to the soil. Also, we have the root samples. Finally, I did the DNA extraction, and we used my 6 sequencing platform to see microbial communities. Here is our initial results that shows on the left side. You can see our phenotypic data, and again you can see F1 hybrids accumulate the threefold higher above ground. This is supported by our fresh sample and then the right samples here. You can see in the bar graph twofold higher root biomass than their parents. Also, we checked our genotypic data. Here, PCI coordinate analysis show the soil and rhizosphere have distinct communities. Also, the hybrids tends to cluster together. The relative abundance analysis shows the rhizosphere and soil phylum for the bacteria. Here, especially for the hybrids in the rhizosphere, we can see the distinction between the hybrids and parental lines for the macrobiomes. Also, to check how diverse are those macrobial communities for rhizosphere and soil, we did the Shannon entropy analysis, and it shows statistically rhizosphere and soils are having the different macrobial communities. However, even if hybrid and parental microbes are different, we couldn't find the statistically significant results. However, these are just our initial data. We need more data to support our idea. With that, I would like to thank you, my advisor, Dr. Chan, and our collaborator, Younger Lab. Also, I want to thank our colleagues, Ryan and Albina, for helping me to making the experiment and data analysis. A special thanks to the Energy Institute for supporting our projects. Thanks you all for listening to me. I'm happy to answer questions.
Question? Yeah. No. Yeah, come on up. John, you can line up over here. If you...
Hi, nice talk. So I'm not a biologist, but from the storage or sequestration aspects, what's to prevent in decomposition a lot of this CO2 to then be readmitted to the atmosphere, or in the anaerobic settings, methane being released? Is there kind of thoughts on, because the carbon's going into the root mass, right?
Yeah.
And then I guess what's the stage beyond that to prevent it from just decomposing?
Yeah, because the roots are using the carbon dioxide to make more biomass, and then our purpose using this approach to make more biomass to store the carbon in the below ground.
Okay. And so that biomass, like, is there any studies of how much of that might break down and reemit CO2 versus how much will stay in the soil?
I'm not sure about that. Actually, I need to check in. Yeah.
Thank you.
Thank you. Yeah.
Hey, thanks for that talk. So the MB sample looks like it's got the largest mass of roots, or is that biomass total? The yellow bar?
Yeah, those are the one of our hybrids.
Mm-hmm. And the rhizosphere in the soil, genetically, it looks like is not too different between all of the samples. I mean, maybe slightly different, but can you just speak to if you think most of the CO2 is going to be captured by the plants or by the soil microbes, and what's the relative amount of CO2 you're expecting between them?
It is a great question. Thank you. So we are trying to actually understand the relationship between the specific plant genotype, how they are affecting the macrobiomes. We see that the soil and rhizosphere have distinct macrobiomes. That's what we are trying to show. If the different plants has accumulating the different macrobiome. Yeah.
Thanks.
Thank you.
Yeah, come on up. One more question.
Thank you. I'm just curious why you selected these species. I understand the corn might be significant because, I mean, there's other, you could feed people with it. I don't know what the other plant is. Is there a specific reason why these were selected?
Yeah, because the corn is the one of the plants that show really hybrids, vigor, the phenomena. So once we make the crosses, they are more vigorous than their parents. Also, these are the model system that commonly used in the plant biology, especially for the Arabidopsis and maize in terms of the hybrid vigor. Yeah, thank you.
Thanks so much.
Thank you.
All right, so we have one more talk before the break. Shivajit Bhattacharya, lab to field scale, time-lapse seismic monitoring for carbon storage.
Thank you everyone, and thanks to the industry sponsors of these wonderful projects that I've been hearing for the last couple of hours. So this project is about developing a method to verify the efficiency of seismic methods for monitoring where does the CO2 or hydrogen goes under the subsurface, right? So there are many companies, there are many big and small companies that are involved in carbon sequestration projects or hydrogen storage projects. They're injecting these gigatons of gas or supercritical stage under the subsurface formations. And they want to know where does the CO2 molecules go. The good news is there are good methods in geophysics that are well-tested methods that people have used to monitor that over time. And here is a result from offshore Norway, from the Sleipner field, where they have collected seismic data over different time periods, and that shows how, with the growth in the CO2 injection volume, how that could change the seismic amplitudes. So you can see those with more injection of CO2 plume, how it grows spatially, and you can visualize those on the seismic cross-sections. That's great. But the bad news is that this method doesn't always work good at every place. The results vary from one site to another. That depends on that because of the survey repeatability. That's a big problem in the geophysics community. That can include your the tropical storms that you have, how it saturates the soil. That can influence your seismic responses. That becomes seasonal, right? And then we have uncertainties in the CO2 and fluid flow dynamics. We cannot see where does the CO2 go. What we are doing here, we are proposing a method, a lab scale method, to simultaneously visualize the CO2 plume and collect the seismic data at the same time. And we can then scale it up to the commercial scale. So here is a lab scale setup that we have, and here is a sand tank. So what we do here, it's a 60 centimeter by 60 centimeter tank. We put different kinds of sand with different grain size, clay, and mix with the water on the top. And then what we have here are these transducers that we immerse in the water, and then with the spectroscope and this acquisition setup, we can automate this data acquisition simultaneously. So we inject air or CO2 through this pipe here, and the CO2 goes here. Sometimes it gets trapped, sometimes it leaks through those layers, and we can image with the seismic data. That's the basics. So here is a result from one of our experiments. So on the left side, we are seeing a seismic section with amplitudes without any air being injected. And when we injected the air and CO2, that's how we changed the seismic amplitudes. We did this experiments multiple times with the different volumes of air injected into the sand tank, and that's how it looks like, how the root mean square amplitude changes with the injection of air volume. So mostly it's a linear change with different injection volumes. one of the interesting things we found that over time, there is a degradation of the seismic data quality. And we now know what's happening in the system because after certain times, what happens, even if you can inject more and more air or CO2, the amplitude doesn't change more. So then the problem becomes, how do you quantify the CO2 saturation at a given point at a given time? Because your 600 tons of CO2 versus 1,000 tons of CO2 will have the same seismic signature. That's a problem, right? So what we did then, we wanted to see that where does the breakthrough happen? Where does the leakage starts happening? So in a kind of anticlinal scenario, it's a pretty common scenario in oil and gas exploration. So we built these two thin clay layers, those black layers that you see here. These are like baffles that are going to prevent the CO2 migration or leakage. And then you start injecting CO2, and here is the gas saturation. So at the top, you see, there's a higher gas saturation than at the bottom. But when you start injecting more and more of this gas, there is a point when it starts a breakthrough. There's a leakage starts happening from this system. These things also happen in the subsurface. So what we are doing, we're collecting the seismic data and finding out what's the limit of the seismic detection limit where you can start seeing this leakage. So there will be more and more experiments will be ongoing. And thanks to the sponsors again, thanks for your support and help.
Questions? Yeah, come on. Oh, wait, hold on. I'll bring the microphone to you.
On the Norwegian study, what were the depths on that, and how much are in your porosity and permeability breakthrough there? How much of that is tied to depth?
So you mean in the study in Norway? Yeah, so Norway study, I mean, they are injecting the CO2 in the porous and permeable sandstone reservoirs that has more than 20%, 25% porosity and pretty high permeability. Depth-wise, it's a couple of kilometers or several thousand feet below the surface.
Yeah. So in your studies, is there any consideration for any reactivity that's happening underground? Like, is it reacting with the sandstone at all?
No, in this case, I mean, that depends on your time for these experiments and the scale of these experiments. So at this lab scale and the time span that is involved, we did not see any reactions or even dissolution. But what we know from many of the commercial studies, I mean, in generally, it depends. I mean, some sandstones that contains a lot of feldspar, there could be some reactions. Some areas we may not see that. If you're in a carbonate reservoir, that can be different, right? So it depends on your injection rate and many other factors, and the time of injection, how much time you're having for this monitoring. Can you see those results or not?
Other questions? I'm curious if you can use your method to detect leakage. Can you detect where the CO2 is, if it's, migrating towards the surface or anything?
And that's the plan. So we are actually, we'll probably submit another Energy Institute proposal next year on this topic. We are working on that, and mostly for the CO2 or hydrogen, especially through the leaky wellbore, because that's becoming a problem, because many areas you go and want and have a really good information about the subsurface, and you want to inject the CO2, but there are hundreds and thousands of wells that are legacy wells. How do you make sure that there's no issue with the well integrity? So we are kind of thinking about a system that probably will do some experiments, but that will be a little outside the scope of this project next year.
Yeah.
You said that the amplitude stops changing at a certain saturation or increase. What sort of saturations are resolvable?
So we found, it's a very good question, and again, it depends on the scale and what are the assumptions of the boundary conditions. So, and depends on the rock physics, right? And whether it is uniform saturation or patchy saturation. So what we find out that generally, of course, if you're like on the two ends of the spectrum, like you have a 0%, like a baseline condition and 100% CO2, of course, you will see differences. But when you're somewhere between, let's say, 40% to 60 or 70% CO2 saturation, we don't see much of the changes in the seismic amplitude in these experiments and also based on our rock physics modeling. But that can change if we change the rock physics condition like the rock types or porosity permeability. So that can change, but there is definitely a window where you cannot really discern the effect of this fluid saturation from seismic data alone. So that's what necessitates us to look at other methods like multiphysics, like electromagnetic or resistivity methods that are more directly connected with the fluid saturation, right? So we have to integrate multiple other geophysical methods to verify those results.
Okay, one more question, one last question.
Your experiment or your investigation you've been doing with sand, but not with a consolidated rock. Have you tried to do with a consolidated rock, like use a core or something, because for sure it will change all the result. So do you consider to use a real rock, not a gravel or something that you're compacting? You follow me?
So that's a good question. So there are alternate methods people use, they call it digital rock physics, where they have a core flooding, and you can put multiple sensors, and then you can see how the CO2 is or the plume is going through that. But in that case, you are seeing only one dimension, right? Here you can see both X and Y, right? That's one of the differences and the value proposition. So it's not necessarily about exactly where the CO2 is moving at one given time. We want to see, can these methods really detect that CO2, right? The surface-based geophysical methods. And what we see, sometimes they can and sometimes they cannot.
All right, great. Let's thank the speaker again.